Good morning. Today we'll be covering the new chapter on nucleic acids. So there are two types of nucleic acids, primarily DNA, which stands for deoxyribonucleic acid, and RNA, which stands for ribonucleic acid. So I'll just briefly try to tell you the main differences between DNA and RNA, which are very important from exam point of view. So you need to know that DNA is a double-stranded structure, whereas RNA is a single-stranded structure. Now, we talked about these bases before. However, I'll just give you a quick recap that there are primarily four bases: adenine, guanine, cytosine, and thymine. Each base specifically pairs with its own base. So, for example, pairs up with guanine always, and cytosine always pairs up with thymine. Now, the difference between DNA and RNA is that although adenine and guanine pair similarly in RNA as well, however, there is no thymine in RNA. There is only uracil. So cytosine has to pair up with uracil. Now, what is deoxyribose? By obviously, deoxy means that an oxygen molecule has been removed. So in this figure, which you can see, the one we can first we will have to know that what is a nucleotide. A nucleotide is a combination of a pentose, meaning a 5 carbon sugar, a phosphate group which is attached, and a nitrogenous base. So you can see that a deoxyribose has one less oxygen compared to ribose. Ribose has 5 oxygens, and deoxyribose and has 4 oxygens. Now, the second important thing is we discussed here that the definition of a nucleotide. So now, how does it differ from a nucleoside? The difference between a nucleoside and a nucleotide is that a nucleotide has a phosphate group attached to it. However, a nucleoside does not have a phosphate group attached to it. Now we need to know the difference between a purine and a pyrimidine. You just need to remember that purines always have double rings, I mean two rings attached to them. And the principal examples are like adenine and guanine. And the examples of pyrimidines are cytosine, thymine, and uracil. As you can see that they all have only single rings, whereas purines have double rings attached to them. This covers purines and pyrimidines pretty much. Now we'll be talking about the four. This is just basic stuff that the phosphodiester linkage. I just the linkage which covers which basically links the two nucleotides. For example, the pairing between cytosine and thymine. Now ATP. What is ATP? ATP stands for 8-adenosine triphosphate. We studied in the chapter of respiration that principally what is the function of respiration? The function of respiration is to use the food we basically metabolize to generate energy, and energy is always generated in the mitochondria in the form of ATP. ATP has, as you can see, three phosphate bonds attached to them. Now, these phosphates are extremely high energy, and whenever they are broken up, broken up whenever the body requires energy for its main metabolic reactions or in the body. Now I told you the differences between DNA and RNA previously. Now we will be studying about the various processes in which the body converts either DNA to RNA and how the two process of transcription and translation occur. Now, what is transcription? Transcription is the conversion of DNA to RNA. So, as I told you that the DNA is double-stranded, whereas RNA is single-stranded, DNA, which is double-stranded, is converted into single-stranded RNA in the process of transcription. Now, how does it work? Now, there are three types of RNA. Messenger RNA, as the name suggests, that what is a messenger? A messenger is a person who conveys a message of a particular person to another person. So messenger RNA similarly acts like it conveys the message from the nucleus to the cytoplasm. It just conveys the message. This is the function of the messenger RNA, and the things you have to remember are that its length is always variable, and it doesn't have a fixed length. Now, what is the function of the transfer RNA? Firstly, you have to remember that the transfer RNA is the smallest in size and the most important, easily the most easy thing to remember is that the name of all three of the RNA suggests their function. So now what is transfer? A person who transfers an object is like he carries an object from one point A and delivers it to point B. So the transfer RNA works in a similar function. It just transfers the amino acids from one part to another part. And there are similarly, since we talked previously that there are 20 essential amino acids in the body, there are at least 20 types of transfer RNA in the body. So here you can see how the transfer RNA basically transfers an amino acid from one part to another. And finally comes the ribosomal RNA. You need to know that the majority, about 80% of the RNA present in the body is the ribosomal RNA. The ribosomal RNA is sort of the synthetic machinery which synthesizes the transcription, and it is the synthesizes the proteins. So it is almost all the machinery and all of the substances which produce the proteins over here. So ribosomal RNA is responsible for the formation of ribosomes. Now, since I didn't talk to you before about it, we need to know that there are two types of ribosomes, 80S ribosomes and 70S ribosomes. Though we will be talking in detail about it in the chapter about when we will be covering bacteria. However, you just need to know that the 80S ribosomes are in eukaryotes, meaning in humans, and the 70S ribosomes are present in prokaryotes, which are bacteria. Now, these are some of the differences which I told you previously as well about DNA and RNA. And you can see that thymine is present in DNA and uracil is present in the RNA. And the major function of DNA is heredity, and it is the transfer of genes from the parent to the offspring. However, RNA is not involved in heredity; it is only involved in protein synthesis. We talked previously that there are two essential components of protein synthesis: transcription and translation. Transcription is DNA to RNA, and RNA and translation is the conversion of RNA into proteins. Thank you.